This video finishes up Chapter 5. Section 5.2 discusses antibodies and the immune system. All organisms are continually subject to attack by other organisms. These pathogens are identified as foreign invaders and destroyed by the immune system. An immune response is triggered by a foreign macromolecule, often a protein or carbohydrate, known as an antigen. The immune system works by distinguishing self from non-self. And indeed, in autoimmune diseases, there is a disconnect between what is the self and what is the non-self, and the immune system ends up attacking self. There are two types of immune systems, the cellular immune system and the humoral or fluid immune system. This section will focus on the humoral immune system. But in the cellular immune system, our own cells are targeted which have been infected. And the cellular immune system can also clear up virus particles and infecting bacteria, and involves the use of macrophages, killer T cells, and inflammatory T cells. The humoral system targets extracellular pathogens, so things outside the cell. It involves making soluble antibodies. It can keep a memory of past infections, and the key players here are the B lymphocytes and the helper T cells. T cells are labeled that way because they come to maturity in the thymus, and B cells are so labeled because they mature in the bone marrow. Vertebrates fight infections with soluble antibodies that bind to antigens. So antigens are those foreign substances that stimulate production of antibodies. Antigens can include coat proteins of bacteria and viruses, and surface carbohydrates of cells or viruses. Antibodies are proteins produced by the B cells that specifically bind to antigens, and they mark the antigen for destruction. A given antibody will bind to a particular small region of the antigen, which is known as the epitope. And one antigen can have several different epitopes. Antibodies are also known as immunoglobulins, and they are displayed at the surface of B cells. When a B cell encounters an antigen that binds to its particular immunoglobulin, it will then engulf the entire antigen-antibody complex. Then T cells will stimulate the production of more B cells that secrete that particular type of antibody. Most B cells only live a few weeks. However, we do get some long-lived memory B cells that can recognize an antigen several months or many years later. And these memory B cells will help you to fight off later infections. There are 5 classes of immunoglobulins: IgM, IgA, IgG, IgE, this should say IgE, and IgD. They all contain at least 4 subunits, 2 identical light chains and 2 identical heavy chains. The heavy chains are heavier by mass because they have a lot more atoms. The light chains are lighter by mass, hence that's why they're called heavy and light. And these subunits associate by disulfide bonds and non-covalent interactions to form a roughly Y-shaped symmetric molecule. So here is a picture of immunoglobulin G. Immunoglobulin G is the one we're going to focus on. And it consists of 2 heavy chains and 2 light chains. So here the H and the L correspond to heavy and light. And then the light chain is composed of 1 constant and 1 variable domain, and the heavy chain is composed of 3 constant and 1 variable domains. So if we look here in the light chain, here it has a constant domain and a variable domain. In this light chain, we've got a constant domain and a variable domain. In the heavy chains, so here's one of the heavy chains. Here's the second heavy chain. So the C represents constant, the V variable. So in the heavy chains, there are 1, 2, 3 constant domains and 1 variable domain. The antigen binding site there's two of them, are located up here, and those are formed by the combination of the variable domains from one light and one heavy chain. And they're called variable because they do vary. They will have different amino acid sequences. And this variability is important because it allows us to form a lot of different types of antibodies to recognize a huge diversity of different antigens. Notice here also the sulfide, the disulfide bonds connecting the chains. The section up here is called the FAB section, which refers to the antigen binding fragment. This section is called the FC section, which refers to the crystallizable fragment. This image shows the antigen binding sites on the antibody and how an antigen will fit into those particular binding sites to form the antigen-antibody complex. All immunoglobulins have a characteristic immunoglobulin fold, which is a sandwich-like structure composed of 3- and 4-stranded antiparallel beta sheets that are linked by a disulfide bond. So here would be the front view, and this is the side view. So here you see slightly different colors of blue, but you have a 4-stranded antiparallel beta sheet here connected to a 3-stranded antiparallel beta sheet with this disulfide bond. This is a ribbon model of the first complete immunoglobulin G molecule to be crystallized and structurally analyzed. And you can see that the molecule has 2 identical heavy chains, which are shown in the 2 shades of blue, and 2 identical light chains, which are shown in the 2 shades of red. Antigens bind via an induced fit. The FAB fragment of an IgG molecule is shown here with the surface contour colored to represent hydrophobicity. Hydrophobic surfaces are yellow, and the hydrophilic surfaces are blue, with shades of blue to green to yellow in between. So in A, we see a view of the Fab fragment in the absence of antigen, and then looking down on the antigen binding site. B shows the same view, but with the Fab fragment in the bound conformation with the antigen omitted to provide an unobstructed view of the altered binding site. And then C shows the antigen bound to the fragment. In addition to being important in our immune systems, antibody specificity is an important analytical reagent. And it's the basis of several techniques that identify and quantify a specific protein in a complex sample. And a general representation is shown here. First, you coat a surface with antigens, and then you block unoccupied sites with nonspecific protein. Then you incubate with the primary antibody against a specific antigen. And then you incubate with a secondary antibody enzyme complex that binds the primary antibody. Then you add your substrate, and then you form colored product which indicates the presence of a specific antigen. The specific reaction of an antibody with its antigen is used in the ELISA technique to test for the presence of, for example, herpes simplex virus antibodies in blood samples. In this example shown here, wells were coated with an HSV antigen to which antibodies against the herpes simplex virus will bind. The second antibody used is anti-human IgG linked to horseradish peroxidase. And then following completion of the steps, the blood samples with greater amounts of the HSV antibody turn a brighter yellow. Western blotting, also known as immunoblotting, combines the superior resolving power of SDS phage electrophoresis with a highly sensitive antibody binding assay to detect a specific protein in a mixture of proteins. So in this, proteins are first separated by size using SDS phage. Detecting a specific protein by SDS phage alone is usually not possible, but Western blotting provides the sensitivity to detect a specific protein. In this technique, proteins are transferred from an SDS phage gel to a polymer sheet in the blotting step. Protein transfer from the gel to the polymer sheet is done by electrophoresis. In an electric field generated by a power supply, the proteins, which are coated with negatively charged SDS molecules, migrate toward the positive electrode. As the proteins migrate out of the gel, they are captured on the sheet, and the proteins will stick to the sheet in the same position they had in the gel. Thus, the sheet is an exact replica or blot of the gel. The empty gel is discarded, then a primary antibody that will only bind to the protein of interest is incubated with the blot. After a washing step, only the primary antibody that is bound to the protein of interest remains. Several methods can be used to detect the antibody-labeled protein. All of these involve using a secondary antibody which binds specifically to the Fc domain or the tail region of the primary antibody. Typically, the secondary antibody is linked to an enzyme that produces a chemiluminescent or colored product, or it contains a fluorescent tag, enabling identification and quantification of the protein of interest. Illumination with the appropriate wavelength makes that tag fluoresce, and the emitted fluorescence can be detected by specialized detectors. Western blotting makes it possible to find a protein in a complex mixture, the proverbial needle in a haystack. Now let's move on to the last section on movement. Organisms move, cells move, organelles within cells move, and most of these movements arise from the activity of protein-based molecular motors. Large aggregates of motor proteins undergo conformational changes that accumulate into a unified directional force. And in particular, we'll focus on the proteins in skeletal muscle. The contractile force of muscle is generated by the interaction of two proteins, myosin and actin. These proteins are arranged in filaments that undergo transient interactions and slide past each other to bring about contraction. Together, those two proteins alone make up over 80% of the protein mass in muscle. Muscle fibers consist of single, elongated, multinucleated cells that arise from the fusion of many precursor cells. The fibers are made up of many myofibrils, and those are surrounded by the membranous sarcoplasmic reticulum. The organization of thick and thin filaments in a myofibril gives it a striated appearance. Muscle fibers are composed of A bands and I bands, shown here. The A and I bands arise from the arrangement of thick and thin filaments which are aligned and partially overlapping. The I band is the region of the bundle that in cross-section would only contain thin filaments. The A band stretches the length of the thick filament and includes the region where parallel thick and thin filaments overlap. Bisecting the I band is a thin structure called the Z disc, perpendicular to the thin filaments and serving as an anchor to which the thin filaments are attached. The A band, too, is bisected by a thin line, the M line, or M disc, a region of high electron density in the middle of the thick filaments. The entire contractile unit, consisting of bundles of thick filaments interleaved at either end with bundles of thin filaments, is called the sarcomere. In contraction, the thick and thin filaments slide past each other, causing a progressive shortening of each sarcomere. Myosin is composed of two heavy chains, shown here in shades of pink, that form an extended coiled coil known as the tail. And then the aminotermini have globular domains. These are known as the head. So you have the tail here and the heads here. Two light chains shown in blue are associated with each myosin head. Myosin aggregates to form a thick filament shown here. So these are the heads in the filament. The myofibrils also contain thin filaments of actin, so actin is the second protein. F-actin is an assemblage of G-actin monomers that polymerize 2 by 2, giving the appearance of two filaments spiraling about one another in a right-handed fashion. This shows a space-filling model of an actin filament that is shown with shades of red, with one myosin head, which is shown with gray and 2 shades of blue, bound to an actin monomer within the filament. On the right here, we see the thin filaments, which will be composed just of actin, and the thick filament here in the center. So notice here that the I band is just going to be composed of the thin filament. Now look at what happens when there's a contraction. The I bands will narrow and the Z discs come closer together. This is also shown in this electron micrograph. So this would be the relaxed state, this is the contracted state. So notice how this band here, it contracted and got more narrow. The use of ATP will facilitate muscle contraction by facilitating a series of conformational changes to protein structure due to the binding, hydrolysis, and release of ATP and ADP. This shows the actomyosin cycle. When ATP is not bound to myosin, a face on the myosin head group binds tightly to actin. So this shows that tight interaction. However, when ATP binds to myosin and is hydrolyzed to ADP and phosphate, a coordinated and cyclic series of conformational changes occurs in which myosin releases the F-actin subunit and binds another subunit farther along the thin filament. The cycle has 4 steps. In step 1, ATP binds to myosin and a cleft in the myosin molecule opens, disrupting the actin-myosin interaction so that the bound actin is released. So here we have dissociation from the actin. In step 2, ATP is hydrolyzed, causing a conformational change in the protein to a high energy state that moves the myosin head and changes its orientation in relation to the actin thin filament. Myosin then binds weakly to an F-actin subunit closer to the Z disc than the one just released. As the phosphate product of ATP hydrolysis is released from myosin in step 3, another conformational change occurs in which the myosin cleft closes, strengthening the myosin-actin binding. This is followed quickly by step 4, during which the conformation of the myosin head returns to the original resting state, its orientation relative to the bound actin changing so as to pull the tail of the myosin toward the Z disc. ADP is then released to complete the cycle. Each cycle generates about 3 to 4 piconewtons of force and moves the thick filament 5 to 10 nanometers relative to the thin filament. Because there are many myosin heads in a thick filament, at any given moment some, probably 1% to 3%, are bound to the thin filaments. This prevents the thick filaments from slipping backward when an individual myosin head releases the actin subunit to which it was bound. The thick filament thus actively slides forward past the adjacent thin filaments. This process, coordinated among the many sarcomeres in a muscle fiber, brings about muscle contraction. Now, the interaction between actin and myosin must be regulated so that contraction occurs only in response to appropriate signals from the nervous system. This regulation is mediated by a complex of 2 proteins, tropomyosin and troponin. Tropomyosin binds to the thin filament, blocking the attachment sites for the myosin head groups. Troponin is a calcium-binding protein. A nerve impulse causes release of calcium ions from the sarcoplasmic reticulum, and the released calcium binds to troponin and causes a conformational change in the tropomyosin-troponin complexes, exposing the myosin binding sites on the thin filaments, and then contraction follows. In Chapter 5, we learned how ligand binding can affect protein function, how to quantitatively analyze binding data, how myoglobin stores oxygen, and how hemoglobin transports oxygen, protons, and CO2, how antibodies recognize foreign structures, and how muscle works.